say that it's just a plain pleasure to be in such an audience and to start talking about fungi. We had a wonderful introduction into the topic, and my main plan is to go and start talking more about fungi. I've been working with fungi for, I try to count or calculate, and I'm not the best sometimes with these numbers, for more than 20 years, mostly with mycorrhizal fungi, meaning that my focus has been on the fungi and how the fungi interact with the plants. My plan is to take you into a couple of stories about fungi, how the fungi maybe talk to each other, maybe how they interact with the plant hosts or other plants, and perhaps a bit more. So let's see. Okay. There's plenty of fungi in the world. Paco showed that there's plenty of other organisms in the world than just humans or mammals. I should have had here a figure of what's the biomass distribution of organisms in the world, and then I would see that most of the biomass actually is constituting of bacterial biomass. And then fungi are somewhere there, fairly important, but not as hugely important as we might think from when looking from the animal side. I really like this comment or the term in the very introduction about the non-human perspectives. So let's see whether we get into a fungal perspective here. So there's plenty of fungi in the world. There's plenty of fungi with different ways of living. The common way of thinking of fungi might be these fungi that are somewhere in the soil, and then they sometimes make these things that we eat and call mushrooms. Many of them are saprotrophic. See, it doesn't work. So they are in the soil. They, as Paco said, they find their own lunch. They make dead organic material into something that's food for them, and they manage this way fairly fine.
Sorry to interrupt. Is there the problem with the clicker?
I can't see. The pointer doesn't work, so the clicker is
the
fine.
pointer.
Yeah, but
okay.
the pointer just doesn't help me. But we manage without. So then there are other kinds of fungi. There are these fungi which actually need another partner for their life, and these might be symbionts and pathogens. So the symbionts, I treat this term here as fungi growing or living together with systematically, consistently living together with another partner, being mostly beneficial or at least neutral, and pathogens doing the same, but actually doing harm to the partner. And these partners might be plants, such as in the case of mycorrhizal fungi, in the soil, you would have plant roots living together with fungi. The partners could be, of course, the plant above ground parts, like the stems and the leaves. There's plenty of fungi within the leaves and the stems. We call them endophytic fungi or endophytes. These partners can also be humans. We have many fungi inside us and on top of us, and they are mostly either neutral to us or even necessary. Sometimes they go out of hand. Sometimes we have also some pathogenic fungi. And of course, there are pathogenic fungi with other animals, such as insects and so on. So fungi are plentiful and they have many roles. And they are important in the ecosystems. As said, I've been mostly working with mycorrhizal fungi, so a brief walk through the main ways that mycorrhizal fungi live with plants. Maybe the most well-known way or type of mycorrhiza that we know is ectomycorrhiza. So this is ecto, meaning mycorrhizal fungal tissue is visible on the root tips. You see it down there, these brownish or blackish tissues on the roots, which are branching kind of two ways, and many of these fungi are forming the visible fruiting bodies above ground. So many of our edible fungi form the ectomycorrhiza with the trees. Then we have orchid mycorrhiza, which is, as said, the fungi growing together with beautiful orchid plants. Most orchids, almost all of orchids, do have this symbiosis, and it's very important for both partners. The fungi need the plants, and the plants, the orchids, need the fungi, in particular with the beginning of the life, where the orchid seeds are so tiny that they cannot manage themselves at all without fungi. Then we have the ericoid mycorrhiza. So again, we have different types of fungi. They're microscopic in the roots, the same as the orchid mycorrhizal fungi. And the plants are a specific set of plants, such as our blueberries or the ericas. And then we have a mycorrhiza, which is most common, most evolutionary old in the world, the arbuscular mycorrhiza, and perhaps the least appreciated, the least easy to see. It's microscopic in the roots. If you look at the roots, then you might see, if you're using the specific aids, that these roots would be having plenty of fungal structures in them, and why it's called arbuscular mycorrhiza, because they have these very highly branched fungal arbuscules within the root cells. And most plants do form this type of mycorrhiza. When we look at the plant phylogeny on the left side over there, then the blue lines, the blue branches of the plant phylogeny are all forming a arbuscular mycorrhizal symbiosis. So majority of the plants we know nowadays are actually living in the symbiosis normally, and many of them depend on the fungal partners for their daily functions, such as nutrition and the stress responses. When we look at the fungi, then actually on the fungal side, most fungi are saprotrophic. These guys which go out to find their lunch from somewhere out in the nature, and the smaller number of lineages, such as arbuscular mycorrhizal fungi or plenty of individual ectomycorrhizal forming fungi, are then getting their food from the plant. When we look into the space, geographical space, and the world maps, here we have been looking with my colleagues to the European maps and asked where are which type of mycorrhizal plants, and is there any regular pattern over there. We are looking here at the arbuscular mycorrhizal plants, which are growing together with all these grasses and forbs and flowers, and in tropics as well with the trees. Then they are more common further south. So the left side map is showing the arbuscular mycorrhizal plant prevalence with the red colors. Further south in Estonia they are also common, but when we are looking at the regional scale range of proportion of plants, then we also have plenty of other mycorrhizal types among the plants, such as ectomycorrhizal fungi. So when we look at the ectomycorrhizal plant dominance, for example, pine trees or the spruce trees, then they are more common further north or in the colder alpine areas, such as we see here in Switzerland or North Italy. So we have different types of mycorrhizal plants, and of course then fungi around us. They are forming individual habitats, and these habitats change from place to place. The point about the mycorrhizal symbiosis, the interaction, is partly about food. So the fungi are helping the plants, be it a wheat plant or some flower or some bush, to get the soil nutrients from the soil with the help of the fungi into the plant tissues. And the mycorrhizal fungi get the food from the plant. They get the carbon from the plant, and they cannot utilize the carbon if it would be provided them in other ways. So this symbiosis is absolutely necessary for the fungal partners, obligatory for the fungal partners. When measured, and I here stress when measured, we're talking about evidence, then it's possible to find that a lot of the plant nutrients are gained with the help of the fungi. And we can also see that a lot of the plant carbon is spent on letting the fungus to live in the plant roots. This is difficult to measure, and usually we don't have these numbers, so we are making assumptions or educated guesses. Of course, these things also vary in nature. So with this little warm-up about the mycorrhizal fungal life, my intention is to take you to 3 stories, to 3 paths, and the first one is how the mycorrhizal fungi and mycorrhizal plants interact. And the main point is here that the interactions are usually many to many, meaning that many fungi and many plants most of the time in the nature interact with each other. The first walk is into a forest, type of forest which is very common in Estonia. We call it the boreonemal forest, or in Estonian salumets. And for quite a number of years we did study these forest understory plant species. When we look back, then we have these big trees over there. Usually these types of forest also are fairly moist or sometimes even wet. And there's plenty of smaller plants, non-tree plants under the forest understories. And we chose 10 different plant species from these understories. We knew that some of these really have to grow in the forest, so the forest specialist plants. And then we had other plants which actually can grow in the forest or in the grassland or elsewhere, so they are habitat generalist plants. And we were looking at a 10 by 10 meter square. You can imagine how big is this plot, kind of like this room or something. And we collected individuals of all these 10 plant species and wanted to know which avascular mycorrhizal fungi grow with these plant species. You might assume that all of them grow with all fungi are there, all plants are there, so all fungal species and all plant species interact with each other, which was not true. So look at this table just as a kind of like, did the plant species and the fungal species meet? The plant species are rows and the fungal species are columns. And you see that the top 5 rows are finding more or less all of the fungi which were in this 10 by 10 meter plot in the forest. The bottom 5 rows found about half of them only. So the other half of the fungi which were present in this place were not interacting with these plant species. That was something very new for us at that time. We didn't know that it can be so systematic that plants are actually in nature, in natural conditions, choosing, and here I'm using the behavioral term essentially, choosing which fungi to interact with which fungi to grow, even if other fungi are there present. So we found that the forest habitat generalist plants, they actually were much less selective towards the fungal partners, meaning they could have many partners at the same time. And the forest specialist plants were rather selective with which fungi they would live with. And I remind you that in every single case, the same plant individual usually had 10, 15, 20, 30 fungal species in the same root system. So there was always a diverse, let's say, a household of fungi in the roots of one plant individual. we worked on with this topic, and of course we wanted to know why. And seems like part of the story of why is in the way how the plants are getting the food, how the plants are distributing the carbon that they gain from the photosynthesis to the roots and to the symbiotic partners, which is showing that the plants can actually make decisions, regulate who they interact with, and regulate how they are distributing their resources. We looked at the same thing in another system, in a grassland system. So Estonians know what is alva. It's a dry grassland on calcareous bedrock, usually quite thin soil, and can be rather dried through in the middle of the summer, meaning that the conditions might be tricky. And here, the fungal part of helping the plants to cope with, for example, the drought stress might come out very useful. These habitats are usually also very plant species rich. So here we took one grassland site in Saaremaa Island and took all the plant species that we grew there. There was 37 of them. And again asked the same question: which plants go with which vascular mycorrhizal fungi? What we found was about 100 fungi in this grassland, and again, some plants had more partners, other plants had less partners. So here each of these bars or columns is a plant species, and the value over there shows how many fungal species each individual had on average. So you see it ranges from, say, 10, 15 to about 30 fungi, a subset of the 100 fungi in the grassland that was found in this location. And we also, of course, wanted to know again why, what's the difference. So we looked at the grasses and the forbs. There was one fern and there was two shrubs. Was there any difference over there? We tested all kinds of different things, like why, again, the plants might be selecting their partners. First way to look at this is to do a network analysis, and this is one of the visualizations of the network analysis. Here, this is network of connections, not the physical network. So in the bottom we have our 100 fungal species, and in the top we have our 37 plant species. It may look like everyone goes with everyone. That's actually not quite true. So one other way to look at this is to ask how specialized or how not specialized are the grasses and the forbs. So the forbs are on the left and the grasses are on the right. And what is measured on the y-axis is the level of specialization. So the higher the number, the more specialized, more picky, more selective is the plant species. And we see that the forbs actually are rather selective. So I've tried to visualize it there that, it might have like one fungal species. In truth, it's not one, it's maybe more, rather like 10 or 15, but it has less partners. The household of fungi is smaller. Whilst for the grasses, the household of fungi for each individual grass, individual or grass species is much larger. They are less selective. They are much more open to different organizations. So again, in a different system here, a grassland system, we saw that the plant species are selectively associating with the mycorrhizal fungal partners. And this depended on growth form, grasses or forbs. We also looked at other parameters of it, but in essence it depends on the plant traits, how they are selecting the fungal partners. Which takes me to the second story, and the second story is the fungi and the nutrients, mycorrhizal fungi and the nutrients, which has led to a common, at the moment fairly widespread perception of mother tree concept, meaning that there is one tree which is supporting its descendants, by providing them with the nutrients and perhaps other benefits as well as information. Let's see what's the evidence for that story. To which degree the plant individuals and the plant species are known to distribute the nutrients among them. Here's maybe a complicated slide, but I try to walk you through this. So when we start from the left side of it, then we do know that there are these threads of mycorrhizal fungi. You might see the network down there under the trees in the soil, which are connecting the fungus and the plant. The mycorrhizal fungus with the plant host, the plant partner, and it may connect individual plants as well. Now there is less evidence of this, whether the mycorrhizal network, the mycelial network, the physical network of the fungus, how frequently it connects one plant with another plant. So we have seen, we usually measure how many fungi there are in the plant system. Yes, these fungi all interact with this plant individual, but it's very complicated to actually identify whether the connection is between plant individuals. So there's much less evidence of that, in fact. But we do know that the mycorrhizal plants usually grow rather well. They might grow better. The more diverse the mycorrhizal fungal community with the plants, the better might be the growth of the trees. And we also might know that the trees can be connected with genetically different fungi. Again, the same species, but the different fungal individuals might be connecting the trees. So there's the evidence again that there are these fungal networks that influence the growth of the mature trees. But how the mature trees are influencing the growth of the tiny trees nearby, that's more of an open question. So when we think of how the food, the nutrients, also the water, might move from a mature tree to a young tree, a seedling, then there are a number of different ways. And one way, of course, can be this fungal network, the physical fungal network connecting one tree to another, or maybe one plant in the grassland to another, in the case of a vascular mycorrhiza. And the nutrients and the information and the signals can move through this hyphal network. But the nutrients and the signals can also move through the soil, because they would move out from the fungal mycelium, move in the soil, and can be picked up in the other plant. And the evidence for differentiating between these pathways, the fungal pathway and the, let's say, direct root-to-root pathway, or root to soil to root pathway, the evidence is not that clear. So the nutrients can move from one plant to another with the help of the mycorrhizal mycelium, but whether this is biologically meaningful in terms of quantities, we actually don't have evidence for that. And also to be able to measure all these things, we need very carefully conducted experiments with controls, with excluding, for example, the access of roots to individual plants, the access of fungal mycelia. So a lot of care should be taken. I'm skipping that stuff. Sorry. So in brief, in the forests, there's plenty of mycorrhizal fungi connecting the trees, bushes, the adult trees and the young trees. It's clear that the nutrients are moving over there. It's also clear that all these individual fungi here, ectomycorrhizal fungi, saprotrophic fungi, ericoid mycorrhizal fungi, they are generating the nutrient habitat. They are generating the soil, they determine how the carbon, nitrogen and phosphorus are stored or moving or getting leaked or being emitted in case of CO2. So they play a big role in the nutrient cycles, but whether they do it directly, helping plant to plant, or whether there is a lot of different ways of how to contribute to the nutrient cycling and the plant growth, I think we need to be really open to the evidence and being critical in a good way of not narrow-minded scientists, but good skeptical evidence evaluating scientists. Let's walk through to the third story, and this is a story of plants and fungi and insects. So the first one was mostly fungi and plants. The second was plants with fungi to other plants, but what about another level of complexity? And this is why I'm getting more into what Paco was telling about. Do the plants communicate? Do the fungi communicate? Do the fungi help plants to communicate? The brief answer is absolutely. There are signals moving within plants. There are signals that can move from one plant to another by the connecting mycorrhizal mycelium. The CMN here is the common mycorrhizal or common mycelial network. There is evidence for that. So when a plant is experiencing something, it usually releases some signaling molecules, so some plant hormones. It could be volatile organic compounds. It could be hormones like jasmonate. These can be in the air, they can be in the soil, they can be in the plant roots, and they can move again with the help of these mycelia to the next plant. And this has been demonstrated in some cases. And this means that when, again, someone is munching on one plant, then the other plant can learn about it. As with the nutrients movement from one plant to another with the help of the mycelial network, again, we need to critically evaluate what is the evidence. Can there be alternative explanations to these volatile movements or the communicative signal movements? So if the signal is in the air, then just from the air the signal can move to another plant. If the signal is in the soil, can a soil be the place where the signal can move to another plant? So these alternative pathways also exist. And of course the situation in nature is complex. There are many kinds of interactions. So even in the soil we have the mycorrhizal fungi, we have the root endophytes, and we have organisms which are eating both of them as well as they're eating the roots. How these signals are moving in the plants, between the different plants, with the help or by other ways than mycorrhizal fungi. There's a lot still to learn. And maybe to conclude with, the main visual I would like you to take is to think that the plants there growing in the nature or in our agricultural systems or even our pot plants like these ones, they usually have these creatures there with the roots, in the roots, on top of the roots, and of course eating them as well. And they do something. They do some things for the plants which are beneficial. They also do some things which are more beneficial for themselves. And they are always dynamic. They can interact plants, interconnect plants now and then. But then when someone eating the mycelium, the connection from over here can be broken up. So life in soil is dynamic, but it influences life above ground a lot. That's it.
Thanks a lot, Maarja. We learned a lot now about fungi. Probably that raised a number of questions in the audience. Microphone is, again, here in this corner.
Hi. I have a question about the diversity of the fungi. You mentioned you measured it in a forest patch and in a grassland patch, but now you also mentioned the pot plants. I'm wondering, do you also know what is the diversity for the pot plants? Is it severely restricted, or is it as diverse as in the natural habitat?
Mm-hmm. My bet is, and this is now an informed guess, that in the potting plants like these ones over here, which we grow in fairly artificial substrate, then the number of fungi which come with that substrate is rather limited. But in principle, it can be very big. So if we're using natural soil, then the diversity of fungi, the diversity of vascular mycorrhizal fungi with the potting plants can be very, very large. The experiments we do in the greenhouse are showing this, that in a short time, the plant can establish itself with, again, 10, 20, 30 different mycorrhizal fungi in the root system. Yeah. So it depends on where the soil is coming from. Yeah.
Paco is there with his question.
Yes. Yeah.
Sorry that they took away your microphone already.
Yeah, that's okay. Yeah, thanks for the talk. Actually, you raised a very important point, which was about with the CARS study, about being careful, taking it easy, because we might be overinterpreting the data. Mm-hmm. There might be another explanation. And I wonder whether we might find similarly alternative explanations when we change the way we focus on the interaction between plants and fungi. In plants, just plants, we always complain, or I always complain about how plant blind we are when we speak of plants and flowers and pollinators. Like the pollinator is the one choosing the flower, as if the flower wasn't choosing the pollinator. Mm-hmm. We speak of the animal as the pollinator as the one being in charge of doing the decision making, which flower I want to land on, right? And here, it seems to me that something similar might be happening when we speak of the mycorrhizal connections, as if it is the trees and the roots, the ones that are deciding which fungi to hang up with, right? And so I just wonder whether we might be able to put up different empirical designs to throw light upon this issue by changing the focus and thinking of the fungi or those networks themselves as being the ones that are bridging in between trees. We speak as if the mother tree and then get the nutrients to the seedlings, and not about the other way around. So what would happen if we shift the perspective? And would that allow us to think of different alternative hypotheses, different experimental designs, when it is them, the ones trying to reach somewhere to connect fungi?
I really liked the way you focused this question. So being a mycorrhizal ecologist, mycorrhizal fungal ecologist, we have been for a long time talking about the fungal perspective or mycocentric view, because in the mycorrhizal research it has been the same story that it's also plant-centric, but come on, these fungi have their own interests. They also have the fitness. They take care of growing, propagating, and keeping the genes moving around in the world. So they actually are not connecting these plant individuals or plant species out of some kind of let's do good for everybody. They have the reason for that. So obviously the fungi, I would love to have a video like you had about these plant roots looking for the way. The fungi, when they start growing in the soil, they are also looking for the way, and they are looking, mycorrhizal fungi are looking for the plant root. and they are actively seeking and looking for the signals and emitting the signals to find the home. So for them, part of their habitat is soil, another part of their habitat is plant root. So they need to find the home to start growing. And at this contact, when the fungus is reaching the plant root, there's a lot of talking going on. The fungal signals and the plant signals, the fungus is, now I'm translating, but essentially the signals are showing that yes, the fungus is here, and the plant is checking, is the fungus okay or not? So the first reaction is that it's not okay, and all these protection mechanisms start going on, the same protection mechanisms as if the pathogen is attacking. After a little while, the plant is lowering the defense signals, and we could say opens the door. Essentially, the fungus is allowed in, but there's clearly an active process from the fungal side over there to actually enter. And this goes either for the arbuscular mycorrhizal fungi or ectomycorrhizal fungi. The plant and fungus are communicating in terms of establishing the symbiosis. And there are genetic systems there where either the plant has been modified, the fungi are tricky to modify genetically, which generate us models where we can test different steps where the symbiosis is not established. So essentially, there are steps where the symbiosis could be stopped, and we will not have mycorrhiza. And both the plant and fungus play a role here. In these experiments, like to ask how the nutrients move, I think we need to ask biologically important questions. It's easy to ask questions like, does the mother tree help the daughter seedlings? But what's biologically important? We need to focus first on the questions that actually help us to explain how the nature works. So clearly, the microcentric focus, the balanced view on the plant and the fungus side on these interactions, I think this is super necessary.
Yeah, thanks.
Someone else? Kaupo, palun.
Is there any evidence that fungi learn from each other as well?
They sense the environment. They send signals. They move around nutrients and signals, and they move depending on, we say, fungal foraging, meaning when fungi are looking for food or for the partner, then they obviously use signals to make these decisions. I'm not aware of that kind of literature to say whether one fungal individual is moving signals to say, to inform another fungal individual that over there is, for example, more food, grow this way, or over there is the plant host, get that way. So, being a critical scientist, I say that I'm not aware of the evidence, but the fungi are able to move the information. If the information can also move from fungus to fungus, then that would be taking the information in, that would be learning.
I have the question of same sort. As we are speaking about signals between the plants, between fungi, how can we distinguish the molecules that are carrying signals, and are there any neurotransmitters involved?
I don't want to start splitting the hair to say like how we define things, but so are there, how do we differentiate the signals is your first question?
Yes.
Mm-hmm.
Yes.
chemically. Yeah, chemically. So it's the same work that you do with the plants. In essence, you need to measure something, and then it becomes the question how you name it. Yeah.
Thank you.
Mm-hmm.
May I add something there?
There is.
Thanks. Yeah, actually, yeah, that's also, for example, chemical, but also electrical signaling. So in the case of electrical signaling below ground, I guess the best tools we have nowadays at our disposal, because it's a very complex pattern, right? So you need to interpret it. So it's in terms of, I guess this is just in plants. It's done, but I don't know whether the same would apply. But basically, developing algorithms, machine learning or deep learning algorithms that, provided the ecological conditions, would allow you to interpret the signal and try to understand what sort of electrical communication might be taking place. But again, I understand that the electrical communication might be just a tiny part of the equation, because mostly will be carried out chemically, hormone-based or whatever.
Yeah, I mean, the electrical part of the signaling is clearly something that the mycorrhizal world, the fungal world, is talking more about now. So it's not only the chemical signals, it's also the electrics. Again, when you have so complex experimental systems, like, I mean, you can even take this one, a small pine sapling and then a couple of fungi, and you need to start generating the experimental controls, then these systems become rather tricky, not even talking about like a forest understory where you have hundreds of fungi and all these plant roots are interacting there. How do you measure the electrical signals over there? Would be cool to do it. But in the experimental systems, yes, indeed, it's that's one way. Yeah.
Yeah. Actually, question for both you and for Paco. These experimental situations, if we are taking plants and fungi to the laboratory, can we be sure that they're acting similarly as they do in the natural environment? How can we mimic this?
Well, we know that they don't. I was yesterday really reading up on this mother tree story topic and the evidence and no evidence. So one thing is that we have a tree sapling in the experimental system. A tree sapling is not the same as a big tree. I mean, the big tree is not just a big sapling, it's something very different. So when we measure something with the babies, we can't say that the adult humans behave the same way. They have grown up and they function already in a new way. That's clearly one aspect. So we know that the experimental systems are providing us some evidence that cannot be directly translated into the very complex natural world. Paco?
Yeah. Well, no, definitely, I entirely agree with you. One thing is for sure, field observations and lab studies, let's put it, we need them both. And of course they will deliver different goods. So one mistake is that, the one assumption that is usually mistaken is that, in the lab you have the tiny, the scaled version of what you want to find out there. And of course it might have nothing to do at all, or might. So you don't know, but there is no other way than keeping always in mind that it necessarily need not be the case, and we have got to do both and wish for the best, right? But you need to compensate the lack of control in the open, the lack of control that you have in field observations, with the type of control that you do have in the lab, bearing in mind that probably most of the phenomenon of interest is being lost by missing the ecosystem that you cannot bring into the lab, right? So it's paradoxically, the more you impoverish the system, the more you learn, but then you don't know what to do, what you have learned, because it doesn't transfer directly to the truly ecological system, which is the one we care about, right? So it's a compromise there, I guess.
So let's remember that why we do the experiments in the lab, it's to actually really understand some mechanisms. So we are making a system as simple as possible, in order to remove everything else, but just focus on this one thing, maybe this one thing of how nutrient moves from one plant individual to the other. Now, when we go back to the nature, then this one mechanism is part of entire story of all other things happening. So there is no more just one fungus and two plants. There is hundreds of fungi, there are other plants, there are bacteria, there is the environment changing, there is the temperature changing, the water availability is changing, there is the day and night. It's the simple system for focusing on a mechanism and then the natural life where we have many mechanisms operating at the same time. Mm-hmm.
Thank you. Thank you, Maarja, for this very systematic overview.
Thank you.
And, no, one moment, please. One moment, please.
Okay.
Biotoopia hopes that for some odd reason, Arne Næss has not crossed your paths yet.
Not yet.
Although I doubt it.
Well, not in that form at least.
Oh,
yeah. Thank you.
That's good.
Thank you, Peter.
Thank you, Maarja.